What's going on, everyone? Let's do some organelle biology. Pretty simple. This is basic bio. So we will do organelles. Can't spell organelles today. Organelle biology or organelle structures. So here we have this microscope image, and we have the art image, and these will be the structures we will talk about. We have the nucleus over here. We have some lysosomes laying around. And so we know the nucleus is where you have the main genome, DNA, RNA synthesis takes place. We have the lysosomes that are important for the intracellular digestion. And then we have some peri perioxiomes. These are important in oxidation of a toxic molecule. And then we have the mitochondria, which is the powerhouse of the cell used for ATP synthesis by oxidative phosphorylation. We have rough ER and smooth ER, and we will learn about what is synthesized in each one. So we have lipids and proteins. Proteins usually in the rough ER, and smooth ER produces lipids. We have the Golgi apparatus, which is for basically like the FedEx or UPS, and they package proteins and lipids for secretion. We have some endosomes, which are also for sorting of endocytosed material. Chloroplast, which we will not cover in this section, is similar to mitochondria. It's for the ATP synthesis by photosynthesis. And then finally, cytosol, where you find most of the metabolic pathways and protein synthesis, as well as the cytoskeleton. So here, pretty simple. You've seen this your whole life. You got the mitochondria, the Golgi apparatus, you have the ER, which is continuous with the nuclear membrane, which is important, as you can see. It comes off of the nuclear membrane. We have the nucleus, nucleolus, the plasma membrane, free ribosomes, perioxium, cytosol, lysosomes, and endosome. So let's jump right into some organelles. Today is going to be the easy day. So, what is the first organelle we'll talk about? The nucleolus. Nucleolus is important, and the structure of nucleolus is that it is a non-membranous component, as the ribosome genes. This is where the ribosomes assemble. The tRNA is cleaved, and then the nucleolus dissipates in the interphase. So basically, we know that the nucleolus is for the ribosome synthesis. It's pretty dense. That's another concept you have to know. It's dense staining because not a lot of, as you can see, we don't see nucleolus here, but it's going to be a dense material inside the nucleus. Has a DNA for RNA, sRNA. Then we go into the nucleus, and the nucleus is, on the other hand, the double membrane. Unlike the nucleolus, which is non-membrane, has a chromosomes or genome, um, and includes that nucleolus, which with the ribosomes. Nucleolus, once again, double membrane pore, has the 23 chromosomes, so you have 22 autosomes and one sex, and then has a genome, basically. That's the big picture of nucleus, and does DNA, RNA synthesis. Next, we will go into the mitochondria, and mitochondria is, like the nucleus, is also a double membrane. And people say that this was the organelle is not a endomembrane organelle. Basically, it came from bacteria, something along those lines. So just know it's not an endomembrane, whereas all the other organelles are endomembrane. And the major function of mitochondria is ATP production by oxidative phosphorylation. So mitochondria is bacteria-like, like I said. So it has a circular genome inside the mitochondria. Has the genes of RNA protein, does ATP synthesis, and the ATP synthesis, if we want to talk about that, works is that you have the pyruvate and the fatty acids that are shuttled to the citric acid cycle. From the citric acid cycle, the NADH basically is used to pump the hydrogen. And with the help of ATP synthase, you convert ADP into ATP. And that is going into the intramembrane space of the mitochondria. And then that gets pushed out to the outer membrane. So that was our ATP and mitochondria. Next, we will do ER. And ER is basically a continuous membrane of the nucleus, or the nuclear membrane. So like I said, ER continues from the nuclear membrane. The major function of ER is synthesis of lipids and fat. So, lipids and protein. And we said protein, smooth ER, lipids, the other way around. Lipids is smooth ER, whereas proteins is your rough ER with the ribosomes on them. And then ER, we have few structures you have to know. You have an ER lumen with its two nuclear membranes. So this one is also double membrane, like the nucleus and the mitochondria, whereas Nucleolus was non-membranous. So as a nuclear membrane, your nuclear pore, and the supporting lamina. And I will show you an image later with this ER lumen. We know that another big concept to know is that the ER area is 20 times other organelles. And once again, the structure of ER, I can just list that real quick and we will do an image later. So the structure is that you have the nuclear envelope, which is your outer and the inner nuclear membrane. You have a nuclear lamina, a nuclear pore, the perinuclear space, the ER membrane, and the ER lumen. Next, we jump into Golgi apparatus, and the main function of Golgi apparatus is proteoglycan synthesis. Proteoglycan synthesis. This is also a membranous structure, so it is a single membrane. Then it's joined with the nucleus. So what it does, it modifies, sorts, protein for excretion, and lipids. So it's a membrane, it secretes protein. So what it does is basically attaches sugar to the protein. So it attaches sugar to protein. And that's what we mean by modifying it. And this kind of modification, we call that glycosylation. And then you can secrete that protein. So Golgi apparatus, basically protein sorting from the ER. It comes in a vesicle to the Golgi. We do some type of modification like the glycosylation, attaches sugar to it, and that way the protein knows where to go. It's like a GPS. Um, and the carbohydrate synthesis that's produced is that it produces proteoglycans and your GAG, your glycosaminoglycans. Um, and then another side component we can look at is that from ER you can go into the Golgi, and from Golgi you can either be sent by endosome into the lysosomes or endosome to the surface of the cell, or from Golgi with the microtubules you can send to the secretory vesicle. So these are kind of all the pathways that proteins or other material can take from ER to wherever it needs to go. Next, we go into lysosomes, and lysosomes we know are acidic intracellular digestion, acidic intracellular digestion. It also has a membrane, a single membrane, and what it does, it kills anything because it has a low pH of 4.5, along with its digestive enzyme. So we have about 40 of these hydrolytic enzymes, such as, I'll just write a few here. So you have your proteases, your nuclease, your glycosidase, lipase, phospholipase, phosphatase, and then sulfatase. So as you can see, they all end in tase or ase, something along those lines. And the major function is basically kill, that respective. So if you have protease, protein nuclease, phospholipase, and phosphatase. Another clinical application to this is that, so clinically, it's important in the lysosomal storage disease, and the one we care about in the oral cavity or in dentistry. Let's worry about Pompe disease. So Pompe disease is a lysosomal storage disease, is that you have accumulation of glycogen, basically. So it's an amyloid protein in a way. So what happens is that you're deficient in lyso alpha maltase, and you accumulate glycogen. So therefore, you have weak muscles. So if someone has weak muscles, Pompe disease due to lysosomes, and so on and so forth. Then we go into our endosomes, and endosomes fuse with our lysosomes for degradation. So you have they are also a membrane structure. They sort material basically. So they chill with a lysosome, and they ingest whatever they have to and shuttle it to the lysosome, to its homey lysosome. Next, we have a pinocytic vesicle. Pinocytic vesicle, as the name implies, eats up anything. So it just eats it up in a vesicle. or digests it in a vesicle. Peroxisomes are also a vesicle, and they're important because they produce H2O2, and that is essential for catabolism of non-essential anything. So your non-essential fatty acids, your non-essential amino acids, or your any non-essential toxic or toxin. So your oxidation of toxins happens in the peroxisome. And another clinical application to know is that you have something called this, which I cannot say. You have this syndrome because you have a protein deficiency in peroxisome. You have a peroxisome protein deficiency. So that gets rid of all the non-essential components. And then lastly, we just go into electron microscopy. So in electron microscopy, you take a cell, you cut it into thin sections, and you're able to see the image like we have right here. So a cross section, so whichever part you're at, and whatever organelles are present in that cross section or the cut. Fluorescence microscopy, you use some type of mercury lamp, label it with fluorophore, and you're able to see the components. And finally, we have Stokes shift, and I won't go too much into detail, just some fact you have to know. So you have this, the graph, the shift, you have absorption on the left, and you have emission on the right. So the absorption component excites, whereas emission is emission. So emission is filters a longer wavelength, whereas absorption, exciting, is shorter wavelength. And I'm still trying to figure out how to remember this, but it's not a big deal. Just know absorption, excitation, shorter, emission, emit long, wavelength filter. Now let's talk about some of these images, and then I will do a quiz kind of scenario where you can write the answers, and then I will post the answers at the end of the video. So let's go into some of these organelles and try to figure them out. So here we have the summary of all the organelles we talked about. So we have the cytosol for the metabolic pathways. We have the nucleus with the genome, DNA, RNA, ER, smooth lipids, rough proteins. Golgi does the sorting modification. In our example, we did glycosylation. Lysosomes degrade intracellular. Endosomes sort the endocyte material, send it to the lysosome. Your mitochondria does ATP by oxidative phosphorylation, and then peroxisomes does oxidation of all the non-essential components. So here you can quiz yourself, and I will post the answers at the end.